Hi everyone, so in this video we're going to be going over one of the most fundamental biological processes, which is photosynthesis. We know that life on Earth is solar powered and that in our plants we have these organelles called chloroplasts. And in our chloroplasts, that's where we are going to have photosynthesis. We're going to have this capturing of light energy from the sun, and then we're going to convert it to chemical energy that is going to be stored in sugars and in other organic molecules. Photosynthesis, it nourishes almost all the living world, either directly or indirectly. And organisms, they have the ability to obtain these organic compounds by one of two ways, either by autotrophic nutrition or heterotrophic nutrition. So if you are an autotroph, you are going to produce organic molecules from carbon dioxide and other inorganic compounds. Our autotrophs, they are the ultimate source of organic compounds for heterotrophic organisms. Autotrophs, they are the producers of the biosphere. Almost all of our plants are autotrophic. The only nutrients that they require are water and minerals from the soil and carbon dioxide from the air. Plants, specifically, they are known as photoautotrophs. They're known as photoautotrophs because they use light as a source of energy to synthesize organic compounds. But we can also have photosynthesis occur in other organisms like algae, in some protists, and even in some prokaryotes. So if you are a heterotroph, you are going to live on organic compounds that are produced by other organisms. Our heterotrophs, they are the consumers of the biosphere. The most obvious type of heterotroph, it's going to feed on other organisms, like animals. Our heterotrophs, they're able to decompose and feed on dead organisms or on organic litter such as feces or fallen leaves, and those are decomposers. Most fungi and most prokaryotes obtain nourishment this way. But almost all heterotrophs, they are completely dependent on photoautotrophs for food and for oxygen. Oxygen is a byproduct of photosynthesis. So photosynthesis, it converts light energy to chemical energy of food. Photosynthetic enzymes and other molecules, they're going to be grouped in a biological membrane that allows the necessary series of chemical reactions to be carried out efficiently. The process of photosynthesis likely originated in a group of bacteria that had infolded regions of the plasma membrane that contained clusters of molecules. So photosynthesis, it likely has an endosymbiotic origin, right? So when we think about it, in photosynthetic bacteria that exists now, we have infolded photosynthetic membranes that function really similarly to the internal membranes of the chloroplast. And what this endosymbiont theory suggests is that original chloroplast was a photosynthetic prokaryote that lived inside of a eukaryotic cell. So our photosynthetic enzymes and molecules, they are going to be grouped inside of the chloroplast. And chloroplast is where photosynthesis takes place. All green parts of a plant have chloroplasts, but our leaves, they are the major sites of photosynthesis. There is about 500,000 chloroplasts per square milliliter of a leaf surface. In our chloroplast, we have a few different structures. So we have the stroma, and the stroma is sort of like a dense fluid that we find inside of chloroplasts, right? So stroma is a dense fluid that is surrounding the chloroplast. And suspended inside of the stroma, we have this internal membrane system of sacs called thylakoids. The interior of thylakoids, they form another compartment called the thylakoid space. And when we have thylakoids that are stacked, it's called grana for plural, granum for singular. So chlorophyll is the green pigment that is inside of chloroplasts. It's located in the thylakoid membranes. It's really important to understand that chlorophyll is inside of our thylakoid membranes. Chlorophyll has a really important role in the absorption of light energy when it comes to photosynthesis. So now we have to talk about chloroplasts, as they are the main site of photosynthesis when it comes to plants. So we already said that chloroplast is mostly found in the green parts. Well, chloroplasts are found in the green parts of the plant, but that our leaves are the major sites of photosynthesis for more plants. So our plants, they have something called the mesophyll. So chloroplasts, they are mostly found inside of these mesophylls. And a mesophyll, it is the tissue in the interior of the leaf. They are going to carry most of the chloroplasts like we see here, and they provide structural support to our plants. Something else that we have in our plants are stomata. Stomata are microscopic pores that we find inside of leaves. So a formula that we commonly see to describe photosynthesis is this one. And in this formula, we have C6H12O6 as a product of photosynthesis. And while glucose is a really common product of it, it is not the only product of photosynthesis. Photosynthesis, it actually produces 3 carbon sugars. And this 3 carbon sugar, it can be used to make glucose. So when we're looking at this formula, we see also that water appears on both sides of it. This is because we have 12 molecules of water that are consumed and 6 molecules that are newly formed during photosynthesis. We can actually simplify this equation by showing only the net consumption of water, and that would look like this. Plus light energy is equal to 6H12O6 plus 6O2. When we write the equation this way, the overall chemical change during photosynthesis, it seems to be the exact reverse of cellular respiration. But both of these metabolic processes, cellular respiration and photosynthesis, they do occur in plants. But chloroplasts, they do not synthesize sugars by reversing cellular respiration, right? So our chloroplasts, they do not produce sugars by reversing cellular respiration. When we write photosynthesis in its simplest form, this is what we get. And when we're looking at this part right here, CH2O, it's representing that general formula for a carbohydrate. So one of the first clues to the mechanism of photosynthesis actually came from the discovery that oxygen given off by plants comes from water and not carbon dioxide. Before we knew this, the prevailing hypothesis was that photosynthesis would split the carbon dioxide and then add water to carbon to make this. There was a scientist named Van Niel, and he challenged this hypothesis. He saw in a bacteria that he was studying that hydrogen sulfide, instead of water, was used in photosynthesis. And these bacteria, they produce these yellow globules of sulfur as a waste instead of oxygen. So he proposed the chemical equation for photosynthesis in sulfur bacteria. And then he generalized that idea and applied it to plants, and he put this as the reaction for photosynthesis. And it took 20 years, but eventually his hypothesis was confirmed. Researchers, they used a heavy isotope as a tracer to follow the fate of oxygen atoms during photosynthesis. And what they found was that oxygen was produced in photosynthesis only when water was the source of the tracer. And they found out that the hydrogen extracted from water, it's incorporated into sugar, and that oxygen is released into the atmosphere. So both photosynthesis and aerobic reactions, respiration, they involve redox reactions. When we have cellular respiration, energy is going to be released from sugar when the electrons that are associated with hydrogen are transported by carriers to oxygen. This forms water as a byproduct. And as those electrons are transferred, they lose potential energy as they fall down the electron transport chain towards that electronegative atom. And the mitochondria, it harnesses that energy to synthesize ATP. Photosynthesis, on the other hand, it reverses the direction of electron flow. Water is split and electrons are transferred with protons from water to carbon dioxide, reducing it to sugar. And because the electrons increase in potential energy as they move from water to sugar, the process requires energy. And the energy that it uses is provided by light. So photosynthesis occurs in two processes, and each step has multiple steps. The first stage is the light reaction stage, and the second one is the Calvin cycle. The Calvin cycle can also be called the dark reaction, or it can be called a light independent reaction. When we are talking about light reactions, it literally translates to photo, and our Calvin cycle translates to synthesis, hence photosynthesis. In the light reaction, we are going to have the conversion of solar energy to chemical energy. In the Calvin cycle, it's going to use energy from the light reactions to incorporate carbon dioxide from the atmosphere into sugar. When we look at our light reactions, our water molecule, it's split, and it provides a source of electrons and protons, and it gives off oxygen as a byproduct. The light that is absorbed by chlorophyll, it drives the transfer of electrons and hydrogen ions from water to NADP+, which stands for nicotinamide adenine dinucleotide phosphate, and that forms NADPH. The light reaction, it also generates ATP by using chemiosmosis in a process that is called photophosphorylation. Therefore, light energy, it's initially converted to chemical energy in the form of two compounds, NADPH, which is a source of electrons, as reducing power that can be passed along to an electron acceptor, and ATP, which is the energy currency of cells. The Calvin cycle, it was named after somebody called Melvin Calvin, who, with alongside colleagues, he worked out a lot of its steps in the 1940s. So when it comes to light, light is actually a form of electromagnetic energy or radiation. Like other forms of electromagnetic energy, light travels in rhythmic waves. And the distance that we have between these waves, between the crests of these electromagnetic waves, that is what is called wavelength. Our wavelengths of electromagnetic radiation, they range from shorter than a nanometer, which is gamma rays right here, to longer than a kilometer, which is known as our radio waves. And the entire range of electromagnetic radiation is known as the electromagnetic spectrum. The most important segment of the electromagnetic spectrum for life is this really narrow band that we have in between 380 and 750 nanometers. And this is known as visible light. So from 380 nanometers to 750 nanometers. Visible light is what is detected as colors by the human eye. And although light travels as a wave, a lot of its properties are those of a discrete particle, which is called a photon. Photons, they are not tangible objects, but they have fixed quantities of energy. We can think of photons like a small energy packet. And the amount of energy that is packaged in a photon, it is inversely related to wavelength. So photons with shorter wavelengths have more energy. And although the sun radiates a full electromagnetic spectrum, the atmosphere selectively screens out most visible wavelength. So the atmosphere filters most radiation, which allows visible light to pass and power photosynthesis. So photosynthetic pigments are light receptors. When light meets matter, it can either be reflected, transmitted, or absorbed. Substances that absorb visible light are known as pigments. Different pigments absorb photons of different wavelengths, and the wavelengths that are absorbed disappear. When we look at a leaf looks green because of chlorophyll, the dominant pigment. Chlorophyll, it absorbs red and blue, violet blue wavelengths. And light, so it absorbs red and violet blue light, and it transmits and reflects green light. A spectrophotometer, it measures the ability of a pigment to absorb various wavelengths. A spectrophotometer beams these really narrow wavelengths of light through a solution that contains pigment and then measures that fraction of light transmitted at each wavelength. An absorption spectrum plots a pigment's light absorption versus wavelength. And the light reactions can perform work with wavelengths of light that are absorbed. So we have in the thylakoid several pigments that have different absorption spectra. Chlorophyll A, that is the pigment that participates directly in light reactions. It absorbs best in these red and violet blue wavelengths, and it absorbs least in the green. We have something called accessory pigments, which includes chlorophyll B. It includes chlorophyll B and carotenoids. And they are going to capture additional light wavelengths. So overall, an action spectrum for photosynthesis, it profiles the effectiveness of of different wavelengths in driving photosynthesis. So when a molecule When it absorbs a photon, it excites an electron. That electron is going to move from the ground state to an excited state. The only photons that a molecule can absorb are those whose energy matches exactly the energy difference between the ground state and the excited state of this electron. Because this energy difference varies among atoms and molecules, a particular compound absorbs only photons that correspond to specific wavelengths. And that is the reason in which each pigment has a unique absorption spectrum. When we have an excited electron, they are really unstable. Normally, they're able to drop to their ground state in a billionth of a second, and in the process, they release heat. In isolation, some pigments, they emit light after they absorb photons, and that process is called fluorescence. Chlorophyll, when it is excited by absorption of light energy, it produces very different results and in an intact chloroplast than it does when it's in isolation. So, in the thylakoid membrane, chlorophyll, it's organized along with proteins and smaller organic molecules in something called photosystems. So, a photosystem is found in the thylakoid membrane. It is composed of a reaction center complex, and it is surrounded by several light harvesting complexes. These light harvesting complexes, they consist of pigment molecules that are bound to proteins. So the reaction center complex, it's an organized association of proteins that hold a special pair of chlorophyll A molecules. Each light harvesting complex, it consists of pigment molecules that can include chlorophyll A, chlorophyll B, and carotenoids that are bound to proteins. Together, the light harvesting complexes, they act like an antenna for the reaction center complex. When a pigment molecule, when it absorbs a photon, that energy is transferred from the pigment molecule to pigment molecule until it's funneled into the reaction center complex. And at the reaction center complex, we have a primary electron acceptor. These primary electron acceptors, they are going to accept an excited electron from the reaction center chlorophyll A. As soon as that chlorophyll electron is excited to a higher energy level, the primary electron acceptor captures it in a redox reaction. Our isolated chlorophyll is going to fluoresce because now there's no electron acceptor. So electrons of photoexcited chlorophyll, they drop right back to ground state. So there are two types of photosystems in the thylakoid membrane. We have photosystem 2 and photosystem 1. Each of them have a characteristic reaction center, which again is a particular kind of primary electron acceptor that is next to a pair of special chlorophyll A molecules that are associated with really specific proteins. Photosystem 2, it has a reaction center, chlorophyll A, known as P680. So the absorption peak for photosystem 2 is 680 nanometers. Something important to note is that photosystem 2 always comes before photosystem 1. These two photosystems, they were named in order of their discovery, but they function sequentially, and photosystem 2 always functions first. So photosystem 1, it has a reaction center, chlorophyll A, known as P700. So its absorption peak is at 700 nanometers. And these two pigments, P680 and P700, they are nearly identical chlorophyll A molecules. Their association with different proteins in the thylakoid membrane, it affects the electron distribution in the chlorophyll molecules, and that is what accounts for the slight differences in light absorbing properties. So these two photosystems, they work together in using light energy to generate ATP and NADPH. So when we are talking about light reactions, there is two different routes that electrons can flow in, and they are cyclic and linear. In linear electron flow, the synthesis of ATP and NADPH is driven by energizing the two photosystems that are embedded in the thylakoid membranes of chloroplasts. When we have light reactions, these electrons, they flow through the photosystem and other molecular components built into the thylakoid membrane. So in linear electron flow, photosystem 2, it's going to absorb a photon of light. Here we go. So it absorbs a photon of light, and one of the electrons of P680, it's excited to a higher energy state. And then that electron is captured by the primary electron acceptor, which is going to oxidize P680 and leave it as P680 plus. Remember, oil rig, oxidation is loss, reduction is gain. It lost this electron, so now it's P680 plus. So the next thing that happens is that an enzyme is going to extract these electrons from water, and it is going to it extracts the electrons from water and it supplies them to the P680 plus pair. This reaction is going to split water into two hydrogen ions, right? So it splits it into two hydrogen ions and an oxygen atom. And that is going to combine with another oxygen atom to form water. And the protons, they are going to be released into the thylakoid lumen. Each photoexcited electron passes from the primary electron acceptor of photosystem 2 to photosystem 1 via an electron transport chain. The electron transport chain between these two photosystems is made up of an electron carrier called plastoquinone, a cytochrome complex, which is a protein called plastocyanin. As our electrons fall to a lower energy level, their energy is used to produce ATP. And as electrons fall through pass through the cytochrome complex, protons are going to be pumped into the thylakoid lumen, which is going to create the proton gradient that is going to subsequently be used in chemiosmosis. Meanwhile, light energy has excited an electron of photosystem 1's P700 reaction center. The photoexcited electron, it was captured by photosystem 1's primary electron acceptor, which created an electron hole in P700, which produced P700 plus. And this hole is filled by any electron that reaches the bottom of the electron transport chain from photosystem 2. So our photoexcited electrons, they're going to be passed in a series of redox reactions from photosystem 1's primary acceptor down this second electron transport chain through a protein called ferredoxin. The enzyme NADP plus reductase is going to catalyze these electrons from ferredoxin to NADP plus. And those two electrons, they are required for NADP plus's reduction to NADPH. And then NADPH is going to carry the reducing power of these high energy electrons to the Calvin cycle. So the light reactions they use the solar power of photons absorbed by photosystem 2 and photosystem 1 to provide energy in the form of ATP and reducing power in the form of the electron carriers carried by NADPH to the carbohydrate synthesizing reaction of the Calvin cycle. Under certain conditions, photoexcited electrons from photosystem 1, never photosystem 2, can take an alternate pathway. It is a short circuit that is called cyclic electron flow. Cyclic electron flow, it is an alternate pathway to linear flow. Basically, what happens is that the electrons, they are going to cycle back from ferredoxin, right, to the cytochrome complex, and from there they continue on to a P700 chlorophyll in photosystem 1 reaction center complex. In cyclic electron flow, there is no NADPH production, and no release of oxygen. Cyclic flow, it does generate ATP though. So cyclic flow, it generates ATP, but it does not produce NADPH and it does not release oxygen. We have multiple living groups of bacteria that are photosynthetic, and they have photosystem 1 but not photosystem 2. So chloroplasts and mitochondria, they generate ATP by the same mechanism, which is chemiosmosis. In mitochondria and in chloroplasts, they use an electron transport chain to pump protons across a membrane. They pump these protons across the membrane as electrons are being passed along a series of increasingly electronegative carriers. This process, it transforms redox energy to proton motive force to generate ATP as protons diffuse back across the membrane. We have some electron carriers, which include the cytochromes that are similar in chloroplasts and mitochondria. The ATP synthase complex of these two organelles, they are very similar also. The differences between oxidative phosphorylation in mitochondria and photophosphorylation in chloroplasts, that is where they are different. In mitochondria, the high energy electrons dropped down the transport chain. They are extracted from organic molecules and then they are oxidized. In chloroplasts, the source of electrons is water. Mitochondria, it uses chemiosmosis to transfer chemical energy from food molecules to ATP, but chloroplasts, they transform light energy into chemical energy of ATP. There is also a slight difference in between the spatial organization of chemiosmosis between chloroplasts and mitochondria, but there are also evident similarities. So the inner membrane of the mitochondrion is going to pump protons from the mitochondrial matrix to the intermembrane space. The thylakoid membrane of chloroplasts, what it does is pump protons from the stroma into the thylakoid space. In the mitochondria, protons diffuse down their concentration gradient from the intermembrane space through ATP synthase to the matrix, which drives ATP synthesis. When it comes to chloroplast, ATP is synthesized as our protons diffuse from the thylakoid space back to the stroma through ATP synthase complexes. Therefore, ATP forms in the stroma, where it is going to be used to help drive the synthesis of sugar during the Calvin cycle. So the Calvin cycle, it actually regenerates its starting material after molecules enter and leave the cell. the Calvin cycle is anabolic. It uses energy to build sugar from smaller molecules. And carbon, it enters the cycle as carbon dioxide and then leaves as sugar. The actual product of the Calvin cycle is G3P. It is a 3-carbon sugar that stands for glyceraldehyde 3-phosphate. Every single time that the Calvin cycle turns, it fixes 1 carbon. For the net synthesis of a single G3P molecule, the cycle must take place 3 times and fix 3 molecules of carbon dioxide. To make 1 glucose molecule, the Calvin cycle has to turn 6 times and fix 6 carbon dioxide molecules. So there are 3 phases of the Calvin cycle. The first one is carbon fixation, the second is reduction, and the third is the regeneration of carbon dioxide as an acceptor. So when we are in the first phase, which is a carbon fixation phase, each of these carbon dioxide molecules, it is attached to a 5-carbon sugar, that ribulose bisphosphate, which stands for RUBP. The reaction, it's catalyzed by RUBP carboxylase oxygenase, also known as rubisco. Rubisco, it is the most abundant protein in chloroplasts, and honestly, it's probably the most abundant protein on Earth. Then the 6-carbon intermediate, it's unstable and it's going to split in half to form 2 molecules of 3-phosphoglycerate for each carbon dioxide fixed. When we get to the second phase, which is reduction, each of these 3-phosphoglycerates, it receives a phosphate group from ATP to form 1,3-bisphosphoglycerate. So basically what happens is that a pair of electrons from NADPH, it's going to reduce 1,3-bisphosphoglycerate to G3P. And those electrons, they are able to reduce a carboxyl group to the aldehyde group of G3P, which is going to store more potential energy. So for every 3 molecules of carbon dioxide that enter the cycle, there are 6 molecules of G3P formed. Of these 6, G3P is a net gain of a carbohydrate. The molecules, they exit the cycle to be used by the plant cell. Only 1 molecule is going to exit the cycle to be used by the plant cell. But the other 5, they are going to be recycled to regenerate the 3 molecules of RUBP that we need. And that is the third phase of the cycle, which is regeneration of the carbon dioxide acceptor. We have a complicated series of reactions. We have the carbon skeletons of 5 molecules of G3P, and they are arranged by the last steps of the Calvin cycle to regenerate 3 molecules of RUBP. In order for this to happen, the cycle spends 3 more molecules of ATP, and then the RUBP, it's now again prepared to receive carbon dioxide, continuing the cycle. So for the net synthesis of 1 G3P molecule, the Calvin cycle consumes 9 ATP and 6 NADPH. The light reactions, it regenerates ATP and NADPH. The G3P from the Calvin cycle, it's the starting material for the metabolic pathways that synthesize other organic compounds, including glucose and other carbohydrates. Neither the light reactions nor Calvin cycle alone can make sugar from carbon dioxide. Photosynthesis, it is an emergent property of the intact chloroplast that integrates both stages of photosynthesis. This is a summary of the 3 phases. So for the last section of this lecture, we have alternative mechanisms of carbon fixation that have evolved in these hot and arid climates. So, one of the major problems that faces terrestrial plants is dehydration. There have been metabolic adaptations that are going to help reduce dehydration, but they often require trade-offs with other metabolic processes, especially photosynthesis. So, in a plant, we know that the stomata, they are the major routes of gas exchange, carbon dioxide in and oxygen out. We also know that stomata, they are actually the main site of the evaporated loss of water. So when it is a hot and dry day, the plants, they close their stomata in order to conserve water. When the stomata is closed, carbon dioxide concentrations in the airspace within the leaf decrease, and the concentration of oxygen released from the light reactions increases. These conditions inside of the leaf, it favors this really wasteful process called photorespiration. Photorespiration, it is a process in which our plants consume oxygen and release carbon dioxide instead of fixing carbon dioxide in the Calvin cycle. It reduces the efficiency of photosynthesis, but it can also protect plants from damage caused by excess light and metabolic imbalances. In most plants, when I say most plants, I'm talking about C3 plants. In these C3 plants, the initial fixation of carbon dioxide, it occurs via rubisco, and it forms a 3-carbon compound, which is 3-phosphoglycerate. These C3 plants, it includes things like rice, wheat, and soybeans. When the stomata of a C3 plant closes partially, the carbon dioxide levels drop as carbon dioxide is consumed in the Calvin cycle. At the same time, oxygen levels rise because light reactions convert light to chemical energy. So even though rubisco, it normally accepts carbon dioxide, as we start to have a scarcity of carbon dioxide, rubisco has the ability to add an oxygen molecule to RUBP. When it does that, RUBP, it splits into a 3-carbon compound and a 2-carbon compound, and that is a process of photorespiration. The 2-carbon compound, it's going to be exported from the chloroplast and then degraded to carbon dioxide by mitochondria and peroxisomes. Unlike photosynthesis, photorespiration, it doesn't produce sugar. It actually consumes ATP. Photorespiration, it decreases photosynthetic output. And one hypothesis for the existence of photorespiration is that it's evolutionary baggage. But in some cases, photorespiration plays a protective role in plants. So in C4 plants, the first fix of carbon dioxide into a 4-carbon compound, that is one strategy. We have a unique leaf anatomy that is correlated with the mechanism of C4 photosynthesis. In our C4 plants, we have 2 distinct types of cells, and these cells, they differ. We have bundle sheath cells, and we have mesophyll cells. Our bundle sheath cells, they are arranged in tightly packed sheaths around the veins of the leaf. Our mesophyll cells, they are more loosely arranged between the bundle sheath and the leaf surface. The Calvin cycle, it is confined to the chloroplasts of bundle sheath cells. And we have an enzyme called PEP, which stands for phosphoenol pyruvate. So phosphoenol pyruvate carboxylase, it's going to add carbon dioxide to PEP to form the 4-carbon product called oxaloacetate. So the mesophyll cell, it's going to export these 4 carbon compounds to bundle sheath cells through something called plasmodesmata. And those bundle sheath cells, it strips a carbon from the 4 carbon compound as carbon dioxide. It regenerates pyruvate and then it is transferred to the mesophyll. So C4 photosynthesis, it is going to minimize photorespiration and enhance sugar production. So our C4 plants, they actually thrive in these really hot regions that have intense sunlight. We have another type of plant. It is called a CAM plant. CAM stands for crassylation acid metabolism. And what they do is that they store the mesophyll cells of these CAM plants. They store organic acids that they make during the day, that they make during the night inside of their vacuoles until the next morning when the stomata closes. So, an overview of the importance of photosynthesis. Light reactions, it captures solar energy and then it uses it to make ATP and transfer electrons from water to NADP+. The Calvin cycle is going to use ATP and NADPH to produce sugar from carbon dioxide, and it's going to supply energy, and it also supplies carbon skeletons to plants. Photosynthesis, it stores energy, it stores solar energy as chemical energy in organic compounds. It provides fuel for cellular respiration and raw materials for other processes as well. And when we have an excess of sugar, the plants are going to store it by synthesizing starch. And if you remember, starch is going to be stored in chloroplasts and in storage cells of roots, of seeds, and fruits. Heterotrophs, including humans, completely or partially consume plant fuel and raw materials. So when it comes to a global scale, photosynthesis is the most important process on Earth. It is responsible for the presence of oxygen in our atmosphere, and every single year, photosynthesis is going to synthesize 160 billion metric tons of carbohydrate. So there is no process that is more important than photosynthesis is to the welfare of life on Earth.